In the reaction where we're going from glutamate to glutamine, we utilize the enzyme glutamine synthetase. Now, let's break down the enzyme word first. Glutamine synthetase. First of all, we're going to be synthesizing glutamine. That's why it's a syntha. But what does the synthetase part mean? Whenever we have a synthetase, that means we're synthesizing a new bond, and we require an ATP or ADP. So here, since we start off from glutamate and we're going to be going towards glutamine, we use a synthetase because we're going to form a new bond, and that bond is going to form right over here between this carbon and a phosphate, and we're utilizing ATP. So that's why we use a synthetase. The other enzyme is called a synthase. The reason why we don't use a synthase is because a synthase does not use ATP or ADP. A synthetase, however, will use ATP. So in the first step of the reaction, we can see we start off with glutamate, and glutamate is an amino acid. So it has obviously the amino group, the carboxyl group, a hydrogen all around this alpha carbon. And then for its R group, it has 3 carbons, and at the end it has that carboxyl group. And when we take an ATP, we remove the terminal phosphate of this ATP and we attach it to the carboxylate carbon on the glutamate. Pardon me, we attach it to the oxygen of the carboxylate, not the carbon. So we attach the phosphate right over here. So we can see this phosphate right over here. It's coming from the terminal phosphate of ATP, and it binds to this carbon. Now this is called the glutamyl phosphate intermediate. Now this glutamyl phosphate intermediate can now act as a more reactive molecule, which can pick up an ammonia. Essentially, this phosphate is going to act as a good leaving group, and it's going to leave so we can see that, so we can have our nitrogen bind. So we're still utilizing that same enzyme. So this reaction is happening on the same enzyme, just one step after the other. And essentially, we're going to bring in this ammonium, and this ammonium is going to attach to the carbon of that terminal carbon on this amino acid. So we can see this nitrogen adds over here, and we see this NH2 group. So this is referred to as glutamine. Glutamine is also an amino acid. So these two being amino acids, they only differ in this carbon bond at the end. And that's the reaction of glutamine synthetase. So why is this reaction important? Well, in the body, we can't have free-floating ammonia or ammonium because it can be highly toxic. So our body has glutamate, and when our body picks up that free-floating extra ammonia, so it's not toxically going through our body, and we form glutamine. So another way that glutamine is beneficial is that glutamine also acts as a temporary storage unit where it holds on to that glutamine, where it holds on to that ammonia until it is needed. For example, let's say that we want to synthesize a biological molecule that has ammonia in it. The glutamine will act as that ammonia donor for the synthesis of that biological molecule. So we see this a lot in, obviously, wherever we need nitrogen. So where do we need nitrogens? Well, we need them for certain hormone production. We need them in nucleotides. We also need it for the synthesis of amino acids. So that's where we would see glutamine acting as that nitrogen ammonia donor. And once again, glutamine synthetase catalyzes this reaction. We're synthesizing glutamine, and it's a synthetase because we're creating a new bond and we're using ATP.